Welcome to the next lecture in the series of quantitative methods in chemistry, where we are trying to look at how software could be used towards data input, analysis and also plotting. In the last class we saw how when you are trying to determine the enthalpy change for a given process, how when you make a measurement as a function of temperature, you would be able to determine what is the enthalpy change. There you had a simple linear relationship between the independent variable temperature that you are changing to that of the variable that you wanted to measure, which is enthalpy change. Not always you have a linear relationship, and in today's class we will be taking a look at few such examples. In order to do that, I am going to be invoking some of the basic concepts that we might have learned in kinetics, chemical kinetics. So why don't we take a look at some of the equations that come across. Let us start with the zero order experiment or zero order rate kinetics, and we are always talking about A going to products. In this one is able to remember that the rate of change of reactants is given by the rate constant times A to the power 0, because it is a zero order reaction. So this will be minus dA equal to k dt. This when integrated gives you the integrated rate law, which is A equal to kt plus c at time t equal to 0. We know that A equal to A naught. Of course, there is a minus sign here. So therefore this implies C equal to minus A naught. Therefore A naught minus A is equal to kt. That is, if you plot A as a function of t, you are going to get something like this. If t is the variable and A is the concentration at any point of time, you are going to get a constant line with a slope where the intercept is the concentration at initial time, the slope is the rate constant. In this case, the rate constant has the units of second inverse because it is zero order. Now similarly, for a first order reaction, you are going to have minus dA by dt is equal to Ka power 1. This when you rearrange and do the reintegrated rate law similar to what we just did, this is going to get reduced to ln of A over A naught equal to Kt. So this can also be observed in the fact that if you are trying to plot as a function of time t, let us say you are plotting ln of A. So this can be written as ln of A minus ln of A naught is equal to kt is minus kt. Okay, so what will end up happening? Ln of A is going to be equal to ln of A naught minus kt. So once again you are going to have a straight line whose intercept is ln of A naught at t equal to 0, the slope is minus k, right. So you would be able to determine same with the linearized plot. On the other hand, let us say you are plotting A as a function of t. If you quickly rearrange, you are going to get A equal to A naught power minus e power minus k times t. So once you have this, what is going to end up happening at t equal to 0, you are going to have this as A naught, and as a function of time this is going to go like this, an exponential function. Of course this will not die to 0, it will die to 0 only at infinity. It will get very close to 0 at larger times, but it is going to not be equal to 0 until t reaches infinity. Similarly, for second order it is going to be minus dA by dt equal to Ka square. So this is going to integrate into dA is kt, implies plus 1 over A equal to kt plus c, t equal to 0, A equal to A naught, which implies 1 over A minus 1 over A naught equal to kt. We can quickly rearrange the same way that we did earlier. So this is going to be 1 over A equal to 1 over A naught plus kt, which implies 1 over A is 1 plus A naught times kt divided by A naught. Therefore, A as a function of t is going to be something like this. So therefore, for 0 first and second order reactions, let us try to summarize what we saw. You are going to have A equal to A naught minus kT. In this one, you are going to A equal to A naught times e power minus kT. In this case, you are going to have A equal to A naught times 1 by 1 plus A naught times k times t. If you are plotting A as a function of t, you are expected to get a straight line with the intercept being A naught and the slope being minus k. And in this case, if you plot A as a function of t, you are going to get that dies a 0 at 0. For the first order reaction, if you are plotting A as a function of t, then you are going to get an exponential decay as a function of time, where A reaches 0 only at t infinity. On the other hand, if you plot ln of A as a function of t, you get a straight line. And in this case, what you are able to realize, if you plot 1 over A as a function of time, then you are going to get a curve like that, where at time 0 you get A naught and the slope is going to be equal to t. In today's example, we will be taking a look at simulated data for 0 first order and second order reactions and try to find out how a software could be used to distinguish apart from these different order of reactions. As always, we will start with the spreadsheet program. So if you realize here, data has been provided where you have concentrations as a function of time. Since this is simulated data, data looks beautiful with no noise, but in reality when you are measuring it, depending upon the variable that you use to measure, for instance, if you are getting the absorbance, it is going to have some noise that comes from the spectrophotometer. If you are going to be measuring conductance, it is going to have its own error that comes up. But in today's class, for the sake of clarity, we will not be introducing any uncertainties with any of these measurements, just so that we understand how data can be once again analyzed using a software program. So the data that we are going to be using today is given as a in the format here, where as we have already seen in the previous class how to carefully enter data. So this is a time variable where time increases in a certain unit, and let us say that it is given in the resolution of seconds, meaning that there is 0 seconds, 1 second, and data is acquired every second. And this is one example where the concentration of the reactant changes as a function of this time A, and this is concentration of B that changes as a function of time, and this is concentration of C changing as a function of time. Let us assume these three are independent reactions, meaning that they have their own order, and we can assume that these are the only reactants that are required to form the products. Once again, all that we are trying to do is how software could help us analyze data. So therefore, we are simplifying cases so that you can understand the simplest of case and extend it to any case that you might require in the research that you might end up pursuing. So let us first ask, which is the easiest one to plot here? If it is a 0 order reaction, what is going to end up happening is that when you plot the change of concentration of the reactants as a function of time, the 0 order is going to give you a linear curve. So let us try to do it. If any one of them are indeed 0 order reaction, that is going to be ending up as a linear curve. So I am going to select a bunch of data, and if you are able to realize after a point, after the 10th second, there is no more of A present. So I can actually go ahead and eliminate the data set so that plotting becomes easier. How would we plot this? Is you select column A and column B, then you can go to insert, and then you can go to the window that says insert scatter plot, and you are able to see a scatter plot here. And what you are able to realize that it does appear a little linear. So why don't we also include the other plots of A, B and C? So the way to do it is to select all the 4, then say insert and insert a scatter plot. And what you are able to realize here is that different things happen. So let us try to zoom in to the initial portion of this graph. So the way you can do that is by formatting axis here. So let us say that instead of plotting all the points, we will only plot the first points for the first 20 seconds, and what you are able to realize is that there seems to be a blue line that seems like a straight line. There is an orange line that also appears straight for a little while, but then there is a curvy path that comes after, let us say, 12 seconds. The same seems to be the inference for the gray curve. We will soon understand what different orders they belong to, and one point that we are able to realize is that for any kinetic reaction one has to acquire good number of data points in the time domain so as to distinguish. If we are just taking the initial points, we are able to realize that all this could seem like a straight line, but they actually are not. But of these three, it appears that the first one could be the straight line. So why don't we try fitting it with the trend line. So we say right click, add a trend line. When you add a trend line, what you are able to realize here is that it already fits it to the linear curve by default. It's fitting it to a linear curve, and the best way to determine whether it's doing a good job is by showing the fit. And what you are able to see here is that it appears that it's an ideal fit, meaning that the r square that comes up is equal to 1. You are able to realize the r square is equal to 1. This indicates that the fit is excellent, and what you are able to realize is that there is a certain slope that's associated with it, and there is a certain intercept that's associated. Of course, the intercept is the first data point itself, and if you remember and recollect just a moment back, the kind of equation we wrote for the zero order is nothing but A is equal to A naught minus K times T, and this is of the same form where A naught comes as the intercept and the K comes as the slope here. So what you are able to realize here, this is indeed a zero order reaction, that is the reaction that involves A is a zero order reaction, and the rate constant is 0.1 second inverse. We can try to do the same thing for the orange curve as well, but one has to be very careful doing this. Let us try to do that carefully, and then you can always say add a trend line. When you say you add a trend line, you are also able to get something here, but interestingly enough, what ends up happening, let us take a look at the equation and the r square. You are able to realize the r square for such a fit is very bad. What do we mean by bad? An r square of 0 means it's there is no fit at all data is scattered, there is no correlation between the x and y axis. On the other hand, r square of 1 indicates that the independent and the dependent variable x and y respectively are indeed correlated. On the other hand, if you get something like this where you do get an equation and you do have some points that pass. So for the sake of clarity, let me remove the gray points, and you will realize that the orange dotted line, which is the fit, does not have many points that end up passing through it. Okay, so in order to exemplify this, let us try to show something more. We can say minimum can be set to 0. So what you are able to realize is that the fitted line, that is the orange dots, don't seem to be passing over the orange data points, which indicates that the fit is very bad. The same is indeed reflected by the r square that has come for such a fit. The r square of 0.5 indicates it's a very bad fit. On the other hand, if you take a look at the blue fitted line, you are able to realize that it comes up as a beautiful fit with the r square of 1. Of course, this is synthetic data, so you get r squares of 1, but generally whenever you get an r square of 0.99, you tend to believe this is okay and you move on. Now, let us try to see what happens when you reduce the number of data sets, meaning that let us say you only plot the first 10 data points. What ends up happening? So we can try to fit the one that we found out as the zero order reaction. Of course, the intercept has to be set to 1, not to 0, because we know for a fact that the concentration that you started with. On the other hand, let us try to fit the orange line. Then we can say you display the equation, display r square, and display the intercept. Set the intercept to 1. So interestingly enough, what you end up getting for the other orange data point is that the r square is still good. Oh, this is confusing. What is happening here is that you are taking different sets of data points. For the same data spanning a larger time, you get a very poor fit for the linear line, but the same set of points for a shorter time, you get a very reasonable fit. If you are looking at R square, it's about 0.99. Can we believe this is first order or zero order for that matter? Actually not. If you are fitting something linear to the concentration of reactant linear as a function of time, then it indicates that it's a zero order reaction, but this need not be the case. This just indicates the fact that as you acquire more and more data points, different models can be discerned. Models meaning you have to acquire a given number of samples before you are sure that the data that you are getting can be reliably interpreted. So let's go a little forward. Let's not worry for now what is happening here. We will come back to this question maybe a little later. So first thing that we are able to realize is that A is indeed a zero order reaction. Now let's take a look at what is B. Let's try to plot the concentration change of B as a function of time. So you get something like this. Let's take a look at the data points as well, not just looking at the software. What happens is that the initial data points, there is a significant drop in the concentration. The first one drops about 9.5%, then you are having about a 9% drop, then you are having about a 6% drop, not 6 unit drop, and then you are having about a small sets of changes that starts coming in, right? But as you start going farther and farther, you realize that the drop is actually smaller. And even if you go farther, if you are able to realize for the same 1 second resolution, the fall is much more smaller, right? So this could indicate probably it is an exponential drop that comes up. Why don't we try fitting it to a trend line? The first thing that a spreadsheet program does is to fit it to a linear line, but you are able to realize that the linear dotted line does not seem to pass most of the data points. Okay, if you would like to see, we can take a look at the R square. Okay, this is what we saw a moment back. We see that the R square is pretty bad, meaning that you are getting something like 0.47 or rather 0.5, which is not a good fit at all. And more importantly, rather than looking at the R square number, you are able to realize the fitted line does not encompass most of the data points that you have acquired, meaning that it's a poor fit. Therefore, the model that you are choosing is wrong. So instead of doing this, why don't we try to take a look at what other models are possible. Let's try to fit it to an exponential and let's see what happens. To fit it to an exponential, suddenly what you are able to see is that y is equal to e power minus 0.1 times x, but the R square goes to 1. So this could indicate, it's not a linear fit, it's actually an exponential fit. If you recollect a moment back, we just said for the first order reaction, A is equal to A naught times e power minus k times t. So therefore, this nicely fits into the equation. Since we are starting with 1 unit of concentration, we are able to realize, this is indeed a first order reaction. And of course, since it's simulated data, you are also able to see the fact that the R square is actually equal to 1. And if you see now the dotted line, you are not even able to see the dotted line because all of them are indeed passing nicely through all the data points. So let's expand it and take a look. Or we can try to reduce this again, format axis, so that we can try to see how much we would like to take a look into. What you are able to see, the dotted line nicely encompasses all the data points. Once again, if you recollect the older fit for the linear line, you realize that it went something in a bizarre fashion. Let's see whether we can fit again the same data. Okay, so what you are able to realize is that this seems to nicely fit an exponential equation indicating it's a first order reaction. Let us try to see whether it is indeed true. How do we do this? We can take the ln of the concentration. So we have learnt in this class before that if you take ln of something, then it becomes ln, and I am copying that cell in. So this is going to be a ln of B. So I can go ahead and copy this and paste it for all the lines going until a certain amount of time. So I am pasting it and then we get a set of numbers. So now let us plot ln of B as a function of time. If it indeed was a first order reaction, you are going to get a linear line. You end up getting that. So this can also be fit to a trend line, and you can actually take a look at the intercept that's going to come up. If you remember, you got something like y is equal to e power minus 0.1 times x. Here also you get a slope of minus 0.1 times x, indicates that the k is 0.1 concentration inverse second inverse, right? So what you are able to realize, the same inference that we get through exponential fit, you can also get it through a linearized fit. However, this I would not encourage you to linearize all the time, because there is certain amount of problems that's associated in converting the error that you obtain, let's say in concentration of B, and when you take log of it. We have learnt in this class how error propagates, and that causes an under evaluation of the error, which is not a good idea. But this is just a textbook knowledge that I wanted to exploit and see whether it works out. Okay, now that we have seen reaction A is 0 order, reaction B is first order, natural progression is that reaction C is going to be second order. Let's first try to take a look at plotting reaction B and C and see how different the curves look. If you are able to see, there's a small change that comes up. The blue is the fit for the first order equation that we just did, and the orange dots are the one for the reaction that we are trying to figure out. What you are able to realize is that both seem to have a beautiful progression, and they kind of seem similar. One can always argue that this could be a first order reaction with a slightly different rate constant. Maybe it has a rate constant that's smaller than what we saw. However, one good way of trying to see this, we just saw a moment back, if you are able to plot 1 over A as a function of t, if you get a linear line, then it's a second order reaction. So let's try to do 1 over B and 1 over C. We know for a fact that B is a first order reaction. So if you are plotting 1 over B, it's not going to result in a straight line, but we will do this just for the sake so that we understand what we are trying to do. So similarly, you do 1 over C. We are not using dollar sign anywhere here, just to make sure that all the cells indeed do get changed. Okay, so now we have gotten this. Why don't we plot these as a function of time? So I am plotting 1 over B and 1 over C, and whichever gives you a straight line is a second order reaction, whichever does not give you a straight line indicates it's not a second order reaction. Okay, so there seems to be a problem here, because the fit for blue, or the number of points for blue increases significantly. So once again, as always, we like to take a look at the initial set of data points. Let's take a look at the initial set of data points. Oh, so this is also having trouble because the y-axis is way too much, so we can try to reduce this to something like 10. Okay, so what you are able to realize here, this is what is causing the trouble for visualizing the blue and the yellow points, where the y-axis was overdone there. So now when I zoom in properly, what you are able to realize is that the orange line goes in a certain way, while the blue line starts to deviate very fast, which is known because we know for a fact it's a first order reaction for B, so therefore it's not going to be a linear line, and you're exploding as the t starts to increase, the 1 over B is increasing tremendously. Okay, so now let's try to fit these two data points. I'll actually remove, for the sake of argument, I'll remove the blue data point, and then we can try to format and see in its full glory. And you are able to realize that orange line nicely increases as a function of time. You can add a trend line, as we have always done, to get the linear curve. So what you are able to realize, because you got to set the intercept to 1, because your initial data point for time 0 is concentration of 1 unit, and what you are able to realize here, the 1 over A is equal to 1 over A naught plus KT, and what you are able to realize here is that the y, which is A, and the intercept 1 is actually equal to A naught, which is where you started with, and the K comes as 0.1. In this case, it will be 0.1 concentration power minus 2 times second inverse. So for all the 3 simulations, what I did was to assume the same rate constant of 0.1 units. Of course, the units change: 0.1 second inverse, 0.1 concentration inverse second inverse, and 0.1 concentration power minus 2 second inverse. And what we are able to see is that for a 0 order data, if you just plot concentration as a function of time, if you get a linear line, then it's 0 order. If you plot ln of the concentration as a function of time, if you get a linear line, it's first order. If you plot 1 over A, 1 over concentration as a function of time, you get a linear line, it's second order. And we also try to do the opposite. What happens if you plot 1 over A for first order? You realize that it starts to explode. And same could have been done for the first order when you're doing ln of A as a function of time. What will end up happening is that you will realize for a 0 order that also doesn't fit. We are able to exemplify the fact here that as you start using computation programs or software more, it helps us analyze this data very much more faster. Similar to a spreadsheet, I am also going to be introducing you another way of handling this data, and this way is by using a software called MATLAB. So MATLAB is this program where we had already simulated some of the curves that we saw for the error propagation, also the way we saw the Gaussian fits that came for different numbers that we had obtained experimentally. This is a software which also works the same way like a spreadsheet does. Generally you can give variables. The advantage of this software, however, is that you can also read the Excel sheets that you might have prepared for analysis and write programs such that it can be semi-automated towards your data analysis. I am just going to be giving you a brief introduction of MATLAB. I am sure there are other tutorials available for you to understand how this can be used. So the way you saw in the Excel sheet where a certain column has a certain running variable that we saved it as, here also a similar thing can be done by giving different variables. So let's say we want to define time as something like time in seconds as time underscore s. This is just a variable, you can name it any possible way. So let's just say it's time underscore s. So I want to give it 10 such points. What you are able to realize now, the time is nothing but a row vector which has 10 units in it, basically 1 going until 10. Similarly to the concentration of A, so we can say conc of A, you can do the same thing, and the advantage is you can even copy paste from Excel. What you are able to realize here, the time is a row vector while concentration of A is a column vector. So one can always convert them into the other vector by using a transpose, which I am sure you guys would have learned with basic matrices. If you take a transpose of a row matrix, it becomes a column matrix. If you take a transpose of a column matrix, it becomes a row matrix. We can try to plot now time underscore s in the x-axis and concentration of A. Okay, so it's kind of giving you an error that says the vectors must be of same length. Let's take a look at what is the length of the vectors. You see that concentration of A is a 1 by 11 vector, while concentration of time underscore s is 1 by 10. So this is something not okay. So we can always introduce one more into this. Actually, if you remember, the time that we ended up defining should just have a 0 seconds that go with it. So now if you say plot, you would be able to see the plot. You will be able to see the plot where this is the time axis, this is the concentration, and you are able to see a straight line. Remember, we do not have points between 0 and 1 second, but this software ends up showing this to you. So rather than plotting it this way, you are better off plotting it with data points in this aspect. So what you are able to realize is that there you go, that's the data point that you are trying to look at, and as a function of time, this indeed does change. So the same way we ended up fitting in Excel, you can use a tool called curve fitting tool. If you call this module of curve fitting tool, then it's going to come up with a dialog box that looks like this, where you can say, okay, my x-axis is time, while my y-axis is concentration of A. Then what you are able to realize is that even before we say anything, it fitted it to a polynomial and is saying the polynomial is p1 times x plus p2, where p1 here is the slope that comes as minus 0.1 and the intercept comes as 1, and it's saying within 95% confidence interval, the range is -0.1 to -0.1, meaning that there is literally no uncertainty, and so is the case for the intercept, which goes between 1 and 1, which means that the only value it can take is 1, and if you're able to realize the R square comes as 1, which once again is what we saw even in Excel spreadsheet, and there are also other ways of judging the goodness of fit, so therefore you can understand how good your model agrees with the data that you have generated. So similarly, okay, now that we have done this, can we do the same thing for concentration of B? However, if you remember, we just saw the fact that if you are just taking the initial set of data points, you could fit to something which fits reasonably to the linear plot. However, you will be misinterpreting a first order reaction as a zero order reaction. So that is not something good. So one way of doing it is to read the entire set of data. So how am I going to do it? Is by using this function called XLSRead. So this is a predefined program within the software called MATLAB, which can read Excel files. But before doing the same, the first step that you need to do is to make sure you have the file set in the correct place. So what I am doing here is that I am using the command pwd, which prints the directory that I am working in. So this is like the way you might end up in a Windows computer, you might see it's a C:/Windows/downloads user/downloads, but in this case this is a user by name creates M3, and the files within downloads is where you are sitting, and if you say list the set of files that have the extension XLSX, I am actually using this Excel sheet to do our analysis. So now what we can do is that we can say data is equal to XLSRead, and then you say 2019, the file name, kinetics, and then you hit enter. Then what you are able to realize is that MATLAB is already able to smartly read the different columns as well with the precision that you have set the data set into, right? If you had 4 decimals, it's going to read into 4 decimals. If you had more decimals, it's going to read into more decimals. But what you are able to realize is that it has now converted the Excel sheet into a matrix where the matrix has different lengths. So basically you go until 100 seconds for all of this. So basically you have 101 data points including the 0 second. Now what we can try to do is to see how this data can be accessed. So all of that is present within this variable called data. So if you hit data and hit enter, you are going to get this. On the other hand, you can also go to this workspace where all the different variables that you have are indeed set. So if you open selection of data, you are going to be realizing that the same Excel sheet is what is displayed here. So this is kind of nice, so that you can toggle between a software that you are comfortable with and also use a software which can do few of these automatedly. Now the exercise comes into the fact, how will I define time from this? So basically if you are having 4 different columns, I should be able to pick the certain column that I am interested in. And the way we do it is that we say data bracket open colon comma 1. This indicates just go pick all the elements from the first column. If I hit enter, what you are able to realize is that it's only picking the time axis here. So this is a nice way of handling the data. Now that if your Excel sheet is comprehensive and you have different variables that have been carefully acquired, this will be a very easy way for you to analyze the data. Let's take the example that we took earlier in the class, where we were trying to understand what is the critical micelle concentration of a detergent, and where you had spectrum that was acquired as a function of the surfactant concentration. You are going to get many different fluorescent spectra that can be analyzed the same way, because you started the excitation at the same wavelength and you had the emission recorded between 400 and 725 nanometers. So you are going to have all of that displayed nicely, so you can call in different such files and read and do the data analysis by carefully calling only that given column. So now that we understand, so we can now redefine our time underscore s as that first column within that file. The semicolon here, what it ends up achieving is to not to display this data. If you wanted to display this data, you can always say time underscore s, and you are going to see that now the time underscore s has this variable. Now let's go directly into concentration of B. Now I can say concentration of B is, if you remember, the first column is time, second column is concentration of A, third column is concentration of B. So therefore what we have to do, colon comma 3. So it's actually data, not date. So now what ends up happening is that now concentration of B is saved into from the data colon comma 3, it's now saved into the variable concentration underscore B. Let's once again reopen the curve fit tool. So now what we can do is to set it to time axis and then concentration of B. So what you are able to see is the same set of plot that we saw a moment before. So instead of using a polynomial fit, one can always change it to an exponential fit if it exists, and then try to see what works out, and you are able to nicely see the blue line very beautifully passes through all the data points, and you are able to get the same results that you obtained before. That is, the prefactor of the exponent is 1, and this the B, which is the rate constant that you are getting here with a minus k, is k equal to 0.1. And your fit is beautiful, and you are able to visualize what is going on. So what we have done so far is that if you nicely save your data into a software, it can be recalled into different programs. In this case, I am just showing you an example of a spreadsheet and MATLAB, which is a tool that you can use in order to plot your data. On the other hand, what you are able to realize is that exponential fits are nicely done, and you can have different types of fit that one can do. You can do a simple linear fit and see that the fitting is bad. Actually, this is not even a linear fit. Let's go to polynomial, and then let's say degree of polynomial is 1. You realize that the fit is extremely poor. Let's try to see how a second order polynomial works. Okay, the R square improves from 0.47. Let's take a look at it again. This is exactly the same R square we saw in the spreadsheet program. So it goes from 0.48 to something like 0.82. So although the R square is getting better, you are still able to realize that the points don't fall within the fitted blue line. So this is still not good. Let's try to take 3. Aha, now we are starting to get closer, but you are clearly able to see differences that come. Some of the data points are above the curve and some are below the curve, but systematically they are off. Not systematically, they are actually off on either side of the curve throughout the experiment. The R square has now become 0.9541. Let's keep on increasing and see what ends up happening. When you go to the power 4, the fit starts to definitely get better, but you are still able to realize anomalies that come. Let me expand it a bit. You are still able to see anomalies that come here, where the blue line does not pass through the black data set. The initial points are also not good, and the final point for sure is going farther away. Let's try to see what happens with 5. So as you are able to increase the polynomial, you are nicely able to see what is going on. But interestingly enough, after the data point that you have acquired, you realize that the number is going to negative, and you know for a fact concentration cannot go negative. So one can keep on increasing to higher orders to see what ends up happening. In fact, here you are able to get an R square of 0.9999. So you might start to wonder right now, how is that an exponential fit that I wanted to get is also satisfied by a polynomial fit? This is not surprising. If you do a simple Taylor series expansion of e power x, you get something like 1 plus x squared by 2 factorial plus x cubed by 3 factorial, sorry, x squared by 2 factorial, x cubed by 3 factorial, x power 4 by 4 factorial, and so on until infinity. So this is not a surprise. As you keep increasing this set of numbers, your R square gets better and better, and you are able to get something nice. But unfortunately, your intuitive understanding of what is the fit is completely lost. So this is a very important point for one to understand. Although your model fits your data very well, if it does not have a physical backing of why that model is chosen, you cannot interpret your results. As a quantitative chemist, it is not just your duty to get your data in a reliable fashion with minimal errors, but also to fit it through and do the analysis with a proper background of which model has to be chosen. If you realize, MATLAB is also trying to tell you the equation is badly conditioned, remove repeated data points at try, center, and scaling. So it's also trying to suggest what's going on. We can try to say whether when you increase it to 8, you're starting to get things better, but still things don't improve until you go back to your exponential fit. Here it does not give you any such error, right? So now what we are able to realize is that this type of data analysis can be easily done by using different software, and how if you carefully and in an organized fashion save your data in such spreadsheet or any other format like a CSV comma separated value file, you will be able to analyze this data. I hope this week of lecture has helped you understand how to save data in spreadsheet, how to analyze very simple linear fits within the spreadsheet program, how to analyze some nonlinear fits in terms of the kinetics of reaction using the spreadsheet program for 0, first and second order, and also using MATLAB. As we go to the next week of lecture, we will be using the simple features of the programs in order to analyze our data, to simulate some data so as to understand how we can sample better. On the other hand, also how to analyze our data with something that's a little more complicated than the simple ln with a linear relationship. Thank you very much.